Hi, welcome to another video. Today we're going to be extracting DNA from an orange. I've already peeled it because no one wants to see that on camera, but I'm just putting it into a bag and covering it mostly to the top with just some warm water. And I am going to just crush the orange in the water. As you can see in a second, there's quite a bit of air in the bag, and that was just being annoying. But just breaking it up into tiny little pieces. Just here, letting all the air out. So about a minute into squishing it, I'm just adding a teaspoon of salt, and this just removes the proteins that are bound to the DNA in the orange. And then just mixing it for another minute and getting all the air out. Once I've done this, I'm pouring it through just a jam bag, through a funnel into a beaker, just to filter out all the big bits that will just get in the way. And I was trying to get quite a clean pour, but I think I might have spilt a little bit on the table. So when it's in the beaker, I'm going to add 2 teaspoons of just normal dish soap to it. After I've just squeezed out the bit with all the juice still in, so that it all comes out. Thought I'd put some gloves on for this, it was a bit icky. And the bit still in the jam bag I'm just going to bin. So I'm adding 2 teaspoons of dish soap. Doesn't have to be like a particular flavour. And this breaks down the cell membranes so the DNA can be released. It was quite fiddly measuring out the teaspoons because it just doesn't work. I'm going to stir it and just try not to make any bubbles. It's quite difficult, and the isopropyl will just pop a few of them, but it's quite key to not have very many. Just swirling it around as well does work. Now I'm using a pipette to get small amounts of the orange solution into a few just plastic test tubes. And then do one with the whole beaker as well, just to see how it works. Now I'm just levelling them out to make sure there's a fairly even amount in each. Now I'm getting some isopropyl alcohol that I've had in the freezer for about sort of 10 minutes, and just putting it into each of the things just to make a small layer on top. Now DNA dissolves in water, but it doesn't dissolve in alcohol, and that's why I'm using the alcohol to put on the top layer. So now I am going to leave all of these for 10 minutes, but as you can see there's already a small amount of the white strands that is the DNA rising to the top. But in 10 minutes time that will mostly have finished rising. A few close-ups of the DNA as well. Now I'm getting my pipette and taking out all or most of the alcohol layer and just putting it into one normal container. This is so we can see all the DNA from each of them. And I'm going to try and pick it up on just like a chopstick. It was really hard to get out. Because I think with a smaller instrument to get it out you can just twist it round and it sticks to it, but with this one it just kept sticking to the sides. It also had a hair in it which wasn't particularly good. But yeah, as you can see, there's a small strand of DNA. Now I'm going to do the same with the large beaker. The large beaker does have quite a bit more DNA in it than I thought there would be. Almost a half of my test tube. And again, I was just trying to get it out with a sort of skewer and make it work. And there's just a small little globule on the end. But yes, I hope you enjoyed the video, and thanks for watching.